Good afternoon, ladies and gentlemen, and welcome to Berlin Brains. I welcome you to the event series of the Urania and its partners. My name is Jochen Müller. I'm the host of tonight. And I welcome you to Neuroscience on Stage. The partners this year are the Einstein Center for Neuroscience, that's the partner for today. We also have the Bernstein Center for Computational Neurosciences, the Excellence Clusters Neurocure and Science of Intelligence, the Max Delbrück Center for Molecular Medicine, and last but not least, the Sonderforschungsbereich. I don't even know if there's an English word for this, 1315. Okay, today we present you two scientists from the Einstein Center for Neurosciences. Why don't we do this in the Urania when I say Berlin Brains is an Urania event? Well, since 3 years we go on tour with this. We want to be a bit closer to you guys, right? And we go to different locations. This time we're in the cinema hall of the Zeiss Großplanetarium in Prenzlauer Berg. And you may have seen outside that this is a very special week tonight. So maybe you want to come back tomorrow and the day after, and then the day after, because we celebrate birthday here. We celebrate the birthday of a very special instrument, the microscope. On Sunday, April 13th, it's the International Day of the Microscope. And the microscope has an anniversary this year. Its 400th name year. In 1625, the Bavarian botanist Johannes Faber coined the name microscope, because you see, scope, the small micro things. Before that, people called it flea glasses or something else. Right? So we took this opportunity to, well, celebrate the microscope and what we see with it. So we have a lot of events. Check out planetarium.berlin, what we have an offer for you. But today, again, let me come back to what we have at hand here, Berlin Brains, and we present neuroscientists and their work in an easy and understandable way. So when you think neuroscience is hardcore, yes, in a way, because it's so cool, but no, because it's not understandable. The speakers took quite some effort in preparing over weeks for this presentation to really make it worthwhile and understanding for you, and they will show you in a minute more of that. This is what we say, the DNA of the Urania and our wish. And if you, at the end, say, well, there's still something maybe unclear for you, we have a long time to get engaged with you, a long time for discussion. I would say maybe half of the time, so roughly 45 minutes, the two speakers will speak, and then we have the same amount of time for you to ask questions, discuss with us. We don't go only on tour, we record all the shows since many years on video. You'll find them on YouTube or on the Mediathek of the Urania. And I emphasize this because you can go and watch all the cool videos, but what you can't do when watching the videos is engaging with us. So everybody watching this at home, hi, next time better get your ass up and come here, see the show live and talk to us. Okay, who's gonna blow your minds today? Two wonderful scientists, Melissa Herman and Seulkee Yang from the group Underpinnings of Synapse Diversity from the Institute of Neurophysiology of the Charité. They will talk about their work, about exploring the brain's diverse cell types and communication mechanisms, and explain exactly that. They do this together because the conceptual motto of Berlin Brains is science is teamwork. And that's why we have always two or more speakers on stage. The first usually is the principal investigator, the leader of the work group, or the professor, giving us a broad introduction about the general area of research. And then today, a PhD student taking past pro toto, one project, giving us a bit more insight, also showing us some data. But of course, there's much more. So if you want to know what else is going on in Melissa's group, that's what the QA is for. Later, when you want to ask a question, just raise your hand, and then I will come to you, give you the microscope, yeah. Priming the microphone so that the people watching the video afterwards also hear your cool questions and the even cooler answers. Okay? So, We heard about the topic, we heard a little bit about the speakers, but let me introduce you a bit more detail into the speakers of today. Melissa Herman, she grew up in the United States. She studied biology in the Reed College, a liberal arts college in the wonderful city of Portland, Oregon, where she stayed to do her PhD in neuroscience at the Oregon Health and Science University. She then, I think in 2010, came to Berlin to start working in Professor Christian Rosenmund's lab in the Charité, where she's still today, but now as head of her own group since 2016, I guess. And the group is, as I said, called Exploring the Brain's Diverse Cell Types. No, that's not the name of the group. That's the title of today. I'm sorry, I'm a bit confused today. Anyway, okay. Well, she and her team do exactly that. They study the identity of cells, and that's what she will talk about today. If she's not doing that, she strolls through the wonderful Kiez of Kreuzberg with her family, with her husband and her 2 wonderful sons. Directly after her PhD student, Seulkee Yang will speak. Seulkee is from Korea. She came to Berlin to study here at the Freie University, biology, I guess, also. And she stayed here to do a PhD in Melissa's lab. She focuses on identifying cells with, well, I don't want to tell too much, but with techniques that she will explain in a few minutes. And if she doesn't do that, she enjoys running. She even runs marathons. Did you participate in the half marathon a few days ago? No? But usually she does, right? And if she doesn't do it, well, she's a very sportive person, so she likes exercising and training, I guess because she sits so much on the microscope and does so fascinating work, of which she will tell us more directly after Melissa. So please, put your hands together, give a warm applause for Melissa Herman.
Okay, thank you, Jochen, for that great introduction. I'm Melissa Herman, and I am very excited to be here. This is my first time participating in Berlin Brains event, and actually, come to think of it, really the first time giving a talk in this to this type of audience and in this kind of environment. And I have to say, the last time I gave a talk was at a meeting that was so specialized that every single person was talking about mechanisms that occur at the synapse. And the person following me was a Nobel Prize laureate. So he has nothing on Seulkee. I'll say that, but just to say, this is a different type of talk for me, but I'm very excited to share some of the questions that we're addressing in the group that I'm leading at the Institute for Neurophysiology, and in particular, exploring this idea of different cell types in the brain and how they interact and the mechanisms by which they communicate. So we're sitting here in this audience, and I think it's probably not an uncommon thought that people are having in their head, wondering how the brain works, right? You're sitting here, you're here because you're at a neuroscience talk, so this is probably something you've thought at some point. And I'm going to give you a spoiler alert now and tell you that I'm not going to answer that question, but what I am going to do is try to convince you that in order to really understand this, to understand how the brain works, we have to first understand how the components that make it up. And we're not the only ones who think this way. This is a big movement in neuroscience right now to really understand the key parts that are making up the brain, and with the hopes that once we understand the pieces, we'll understand better how it functions as a whole. So when I talk about the pieces of the brain that make up what the brain is made up of, we can go down in scale. So we have first, the whole brain. We know that it's causing cognition. You understand that because you're watching me and thinking about what I'm saying now. And the next scale down is that we know that this organ is made up of many different cells that are interacting in networks. These networks are communicating by this specialized structure called a synapse, which is what we're mostly interested in today. And these cells have shapes or morphology and physiology or functions that they must undergo in order to be able to communicate with one another, which is their main function. How these functions happen on an even smaller scale level goes down to the molecular. What are the proteins that are in these cells that can drive the different processes that happen? And how are these proteins translated from the mRNA message, which Seulkee is going to talk about in more detail. And then, of course, on the smallest level, this is all encoded by the DNA and the genome that these cells contain. So now we think, we're already thinking now about the brain on sort of the cellular level. And let me just say, this is really where we're going to focus, where I am most interested in this point in neuroscience. And here you can see from this illustration, what we want to point out is that already you see these cells that are here in different colors, and they're different shapes. And of course, this is a vast oversimplification. But our brains have around 100 billion neurons. This is a huge number of cells in each of our heads right now as we sit and listen and try to understand this talk. And of these cells, there are around 3,000 different types. So what do I mean by cell type? You know, these cells we know are different from one another in both their form and their function. And in fact, this is not even a super new concept. And I think this is an appropriate time because of the anniversary of the microscope to talk about someone who is very famous in this field and also deemed the godfather of modern neuroscience, Ramon y Cajal, who noted even 100 years ago, looking through his microscope at human brain tissue and drawing what he saw, that there were clearly differences in the cells that occurred. And he was actually the first to postulate that the brain is made up of these distinct cells that are somehow, didn't know at the time how exactly, communicating with one another. So he could see just by looking that these cells are different, but are they different on the inside? Are they different in their function? These were things that were not yet clear at that point. And this is part of the driving force for the mission of my group, which is to understand better how the communication at very specific connections between cells happens in different types of neurons. So before we can really start to explore that, it's important to understand how a neuron looks and how it functions and its specialized structures that allow it to do so. So a neuron is a cell, and it may be that from your biology course in high school or whenever the last time it is that you took one, you have an idea of a cell as sort of this round structure. The important parts, of course, being you have the plasma membrane that surrounds it, and then a lot of what we call intracellular organelles that provide the energy that are necessary for the cell, and then the nucleus where we contain all of the DNA that's going to be transcribed into RNA and eventually into protein to allow the cell to function. So a neuron has, of course, these structures as well. It needs it. It needs to do basic cellular processes. And these reside mostly up here in the cell body or the soma. of the neuron. So a lot of the stuff, the nucleus is there, that's where you have a lot of the general cell biological functions. But neurons also have to be able to communicate. They have to be able to receive signals, and they have to be able to transmit them. And for that reason, there are specialized structures that the neuron has. Here, you can see the structures called the dendrites. This is where the incoming signals are received by the neuron. Neurons are excitable cells, meaning that they can then generate electrical signals here in the dendrites. And then the axon is the structure that is going to send the signal to the target, to its target cell. And in the axon, this is where we have the generation from the signals that are accumulating in the dendrite into this wave of electricity called an action potential. And the action potential moving down the axon is really the key to the communication. And then it ends here at this point of contact between the what we call postsynaptic neuron, or presynaptic neuron, I'm sorry, the neuron giving the signal, and the postsynaptic target, which is receiving. And this point of contact is called the synapse. This is where really the action that I'm interested in happens. And we might be thinking that why doesn't this just, or what couldn't this just happen by electricity passing from one neuron to the next, right? Just an electrical signal. This one gets shocked, that shocks the next one. That's not, for the most part, I mean, that can occur, actually. There are synapses where this happens. But for the most part, that's not how nature has really devised this to work. The best way for it to work is actually through the release of a chemical called a neurotransmitter. So here at the synapse, neurotransmitter is released, and then it's detected by the target cell. And this is how you get the generation of the signal in the postsynaptic target. So, sounds simple enough. I think that you even see these schemes sometimes on ads for antidepressants and things. So this is kind of clear. But it wasn't always so clear. And actually, the way in which neurotransmitter is released, this is one of the stories that got me interested in neuroscience to begin with, and particularly in this part and process of neuroscience. When it was first noted that a neurotransmitter was released and that there was a chemical transmission that was happening at these synapses, it was proposed by a very brilliant scientist, Bernard Katz, that the neurotransmitter was released in packages of many, many molecules at once. And they didn't really understand how this happened, but he knew that this was likely how this is what was occurring. So then it wasn't until this was back in the 50s, and if I didn't mention that. And it wasn't until around the 80s that we really saw the first physical representation of this idea. And again, important to note that with regard to the microscope, now we can talk about a different kind of microscope, shedding light, so to speak, but in this case, the light is an electron beam on how this process worked. So here we have a team of John Heuser and Tom Rees, and what they did was they did electron microscopy of a synapse, where first here you can see that these small round structures, these are synaptic vesicles. And the idea is that each of them is packaged with thousands of molecules of neurotransmitter. And this makes sense. This fits with the theory that they can be released during neurotransmission. But how does it happen? So what these guys did, which was very clever, was that they could stimulate the synapse with electricity. And then they could freeze the synapse in order to arrest the structures that occur after the stimulation. And what they saw then were these omega shapes, which is now known as the morphological, the structural correlate of the fusion of these organelles, the synaptic vesicles, with the plasma membrane. So this was a way that we could see that with electron microscopy, we could actually see this theory come to life that Bernard Katz had proposed about the quantal, as it's called, this release of neurotransmitter from the presynaptic cell. So not only do we have, so it's important to point out that this neurotransmitter release process and communication by neurotransmitters is not happening with just one flavor of neurotransmitter. There are different kinds, and these may not be even the most famous neurotransmitters or the ones that you've heard about before, because a lot of the ones that are more famous, let's say dopamine, serotonin, these ones are not acting in this sort of fast chemical transmission that the transmitters I'll talk to you about today do. So here we have an excitatory neurotransmitter called glutamate and an inhibitory neurotransmitter called GABA. And these are really doing most of the work of the chemical transmission in your brain that's occurring at any given time. And the thing that is very interesting and important about chemical transmission is that it doesn't have just one direction. So if we think about an electrical synapse, so this is just electricity going from one cell to the next, all this signal can do is to excite the target cell. So you shock this one and then it shocks the next. But the great thing about chemical transmission is that you can have these different transmitters that have very specific receptors that, when they are activated by the transmitter, can open a specialized gate called an ion channel. And depending on what this ion channel allows through, this can either excite the postsynaptic cell, which makes it more likely to have to transmit a signal, or it can inhibit the postsynaptic cell, which would calm it down and make it less likely to transmit a signal. And just from sort of our common, somewhat common knowledge in pharmacology, I can say that you might be familiar with some of the activations of inhibitory neurotransmitters like GABA, and especially their receptors, because there are some very common drugs such as anti-anxiety medication and sleeping pills and even anti-epileptic drugs that act on these receptors to enhance the amount of transmission that occurs at the inhibitory synapses. Right, so I have showed you pictures of two synapses. That is not actually what it looks like in the brain. In your brain, as I said before, 100 billion cells, 1,000 trillion synapses. So there's really a lot of synapses that are happening. And they have to all be organized in a way to drive the activity between different neurons. So in order to illustrate this a little bit, of course, now I'm still with just one cell. This is how we have to take little bites at a time. I wanted to show you another really, really beautiful image that comes from a very recent study from Jeff Lichtman's lab at Harvard, where they've done, again, electron microscopy. But in this study, firstly, they're using human brain tissue, and they've taken a 1 millimeter cubed volume of cortex from human brain. Then they can they fix it or they preserve it, and then they slice it very, very thinly, and then they do electron microscopy on these sections in serial, so that essentially they can go through the entire volume and then recreate their the cells that they see at an unbelievably small resolution. And this is just an example of one of the cells that was recreated from this data set. And it is itself, this is going to be the postsynaptic target that we're talking about, and it is in fact an excitatory neuron. So these are this neuron's dendrites here coming out. Here's its soma again with all the its information from the genome and what have you. And this here is the axon. So this is where the signal will be propagated to go to further targets. And you might have noticed these little spikes here. These are specialized structures onto which synapses happen. And I think you can already notice that there are a lot of them. And one thing they could do with this data set was that not only do they reconstruct the whole cell, they can also annotate the individual synapses that are occurring onto this neuron. And they can see, distinguish by the morphology that they see at the EM level. This is actually a picture of the synapses that they see, and this is the level of detail that they can see. Here you see the synaptic vesicles in the would be one of these, for instance, orange synapses, and then the postsynaptic target. And here we can see excitatory synapses in orange and inhibitory synapses in blue. So one thing that this can show us, I mean, this is probably 1,000 or thousands of synapses even onto this one cell. And they are both excitatory and inhibitory. And not only will there be this one cell that's receiving this, but all of the cells in the network will. And so the point is that what this cell's job is to do will be to integrate the signals that it's receiving, both the excitatory and the inhibitory synapses, in time and in amplitude. And then basically, depending on how much excitation it receives, propagate the signal to its next target. And then that will result in electrical or network activity. And so this is just, again, a simplified schema of this idea where we have a small microcircuit of neurons interacting, where we have a neuron that is providing inhibition, and then neurons that are providing excitation out of the network, but also between one another. And the key is that this information has to stay in a very balanced state in order to not be able to overexcite the circuit, because when that happens, when we get out of balance between the excitation and the inhibition, we start to see pathological pathology coming about. For instance, we can imagine that if we had in this neural circuit a decrease in the strength of these inhibitory synapses without any sort of compensation of the output for the excitatory synapses, we would end up getting too much excitation, and this could result in epilepsy, for instance. So, given all of that background, we can go back to this question that we're trying to address, which is, how do these different types of neurotransmitters, how are they released, or what are the details and the characteristics of their release that is happening from these different types of neurons, and ultimately how does that affect the neural circuits and brain function? So the main techniques that we use to ask this question are these three. So for now, we use electrophysiology. This is the technique that I've been studying or studied from my PhD into my postdoc and continue to really love and use a lot today. Also live cell imaging of fluorescence signals. This has been very important. And then finally, much more recently, we're using a cutting-edge technique called mRNA sequencing, which Seulkee is going to tell us more about when I'm finished. So I'll just go through briefly the first two techniques just to give you an idea of what we are generally asking with these techniques. So the first is electrophysiology, which is really a very fun technique to work with if you want to have a real-time idea of what's happening with your cells. And the way that this works is that we use cells, predominantly cells that are in culture. So we have cells that we can grow in a petri dish and then put them in the incubator. We take them out. This is what a cell would look like on the microscope. Then we can take a microelectrode that's in a glass pipette with a very, very small tip. And we can take this microelectrode and put it into the cell, and using sophisticated electrical circuitry and amplifiers, we can actually record the electrical signals that this cell is receiving when neurotransmitter is released onto it. And this here, which is something that we call a trace, is an example of that. And what we see here is its current over time, and these little blips here are actually the response that this cell would be having to the release of a single package of neurotransmitter, like we saw from the electron micrograph I showed you earlier, and as Katz had proposed back in the 50s would occur. And with this technique, we can look at how GABA and glutamate is released from different synapses. We can look at how the strength changes with different treatments by drugs, and we can see the flexibility and the characteristics of the release from those synapses. Then another technique that we use very often is a live cell imaging technique where, again, we have many, many neurons that are growing on a culture dish, and they're making synapses onto one another. This blur here that you see of the dark blue here would be synapses and neuronal processes, dendrites and axons. But the signal that we see and that we use for these techniques is a fluorescent signal. And we have these specific sensors that we can use that increase their fluorescence or their intensity when neurotransmission has occurred, when the neurotransmitter has been released. And from this, we can learn things about what's happening at the synapse. So I'm going to show you an example here of one of these probes. So here, again, as I said, we have synapses that are in this picture. You can watch it. It's pseudo-colored, so you can see that the dark is where things are dark, and it gets brighter as it gets brighter. And you can watch here for a small S, and when the S appears, this is when we've stimulated the cells, and you'll see that the signal goes up. It then decays, it gets lower again, and at the very end, I flash a substance on that shows you where all of the sensor is in the entire field of view, as we call it. And there. Here you can see, there you saw the S, it went back down, and now it'll flash, and you'll see there. So what we can do with this information is we can compare this increase that we see during the stimulation to the increase that we have at the end, which shows us the total amount of sensor that's available. And we can learn again about the strength and the conditions and the characteristics of neurotransmitter release. But what advantage does this technique have over the electrophysiology that I showed you in the technique before? So one thing that we can do here is that we can actually see what's happening at individual synapses. And this gives us an advantage to really be able to say, okay, this type of synapse is different from this, compared to in the electrophysiology, where we're really averaging all of the possible connections that the cell would have. So with that, we're going to move back to a different level. I mean, we're still interested again in the cellular level. We're interested in cell function and how its identity is, the identity of cells is created in a developing network. But so I'm going to hand the microphone over to Seulkee, and she's going to tell you about her project where we're using mRNA sequencing to address these questions.
So good morning, everyone. So my name is Seulkee, and I would like to continue with this project that we are doing in the lab. And so after my presentation, I would like you to hold on to two things. The first, we have just really more than neurons. We have really cellular diversity in the brain. And second, how single-cell RNA sequencing technology help us to explore this incredible diversity. Before we forget anything that we learned from Melissa, let's just jump right into it. So we just learned that we have over 100 billion of cells, in our brains. And we have 3,000 different kinds of them. 3,000 different unique types with their own special functions. And neurons, unlike me, they are very social. So they like to communicate with each other, and they like to be connected to each other. And now, if you are trying to imagine how many connections a neuron would ever make, the complexity becomes just next level. It's 1/4 billion of synapses that they could ever make. But this raises an important question, because we are scientists. We always have to ask why. So why does our brain need such an incredible diversity? Well, because brain isn't just a simple circuit board. It's not just sending signal from position A to B. It's more like an orchestra, where each instrument plays a unique role in creating the beautiful harmonies. We have excitatory neurons. They're like musicians, so they produce the beautiful melodies of the actions and thoughts by driving the brain activity. And we also have inhibitory neurons, which I'm really interested in, and they're the conductor. So they regulate the rhythm of the whole music and keep everything is in tune by controlling the timing and the intensity of this brain signal. And we also need glia cells. They are the backstage crew members who are like Johan and Felix, and they keep the stage in order by giving the structure support to the neurons. Just like the orchestra needs this balance of instrument, conductor, and crew member to create the beautiful harmony, our brain relies on this diverse cell type to function properly. And who am I? We just saw that how brain is like an orchestra, and we need this diverse cell type to function properly. And as a neuroscientist, I have these two main ideas that I want to understand. First, how does this orchestra develop in mammalian cortex? And second, what happens when something disrupts its harmony? And to answer these questions, I use two main investigative tools. First, electrophysiology, which allows me to listen to the electrical conversation of neurons. And if I notice some differences, like this green and red graph, then I use single-cell RNA sequencing technique to understand what's the driving force of these changes at a molecular level. And now I want to introduce you to what you can get from this technique and why it's such a game changer for neuroscience. And even if you have completely no idea how this technique works, you probably have heard of the heart of this technique, the mRNA, in the context of vaccine. So when mRNA vaccine is introduced to your body, your cell would produce specific protein to get your immune system ready. But this isn't just a trick for vaccine. This is actually how all the cells in your body would operate. And we call this in molecular biology as a central dogma. And this fundamental process describes DNA being transcribed to mRNA and mRNA being translated to protein. But let's just break it down in an easier way so that we could all understand. So DNA is the cookbook. Because I'm Korean, I just put the Korean cookbook. And I probably have in my blood. So DNA is the master cookbook, which contains all the recipe that your body would ever need. And every cell in your body has exactly same cookbook. And mRNA is the temporary copy of the specific recipes that your body needs right now. And the protein is the finer discs that are made from these recipes. And the key idea here is that every cell contains exactly same cookbook, so DNA, but each cell copies different recipes, so different mRNAs. I want to give you more example to understand it in a better way. So now if you were to compare these two images, so they are from different cells. And if I were to tell you to compare them and identify their cell type, you probably cannot do this. Because just by looking at them, they have more similarity than differences. And now if we were to zoom in and look at their mRNAs, you see that the cells on the left, they are the cells from our heart, because they are copying actively the important mRNA for the muscle contractions. And the cells on the right, they are glia cells from our brain. And they are making, we know that they are glia cells because they are making the mRNA that are important to support neurons. So now you can understand why we look at mRNA when we want to understand and study the cell type diversity. But the challenge is that we actually have the mixture of diverse cell type in our system. And they even look different. Like if you were to compare them just by their look, they also look completely different. And they are probably making completely different mRNAs. So if you were to understand or answer questions to like which cell type is affected by the treatment and which cell type carries burden when disease happens, if you were to answer this type of question, you have to look at mRNA at a single cell level. And I want to emphasize this one more time. So when we want to study and understand the cell type diversity, we have to look at the mRNA at a single cell resolution. So now you all kind of like little bit get like what you will get from this single cell RNA sequencing experiment, and imagine that your result will be something like this. So every recipe is somehow organized and they're very structured, so you will know, okay, this is a tomato pasta, spinach pasta, so it's a vegetarian cell or something like this. But in reality is that this is the more or less what you get from this experiment. So we have a just giant table with a lot of different numbers. And each row contains the information of individual cell, and each column has the information of individual mRNAs. And inside we have different number, meaning that how many of that particular mRNA is present in that cell. And really, the problem is that we have 24,000 cells and 15,000 mRNA. So I cannot just look at this and say, okay, cell 1 has this mRNA, so it must be this cell type. No, I can't do that. And this will take me this will take my PhD forever to finish. And that's why my best colleague, my best friend, the AI comes in. So AI is really good at recognizing patterns. And just what we just saw is a very, very complex table. And now we just reduce it in a different way. We just reduce the complexity. I'm not going to explain how this works because I also don't know so much. But if you have a specific question, I can answer it later on. And just for now, we just need to understand this individual point is an individual cell. And they have somehow different color. And they have different color based on their grouping. And they could be grouped together when they have a similar mRNA. So we will have, from this 15,000 different mRNA, we will probably have some relevant and important mRNA, but we will also have some, irrelevant, some random stuff. And then we just trash them, and then we just contain these important ones and put it in this like two-dimensional plot. So let's imagine this is like the magic, how it works. But still, we don't know that which group belongs to which cell type. So this is where the AI friend comes in, and we have first reference data. And here, the individual, each group is already like annotated per cell types, because we know that for this blue cell, for instance, they are called paralbumin neurons, and they need paralbumin ERBB4, LHX6 to be identified as paralbumin neuron. So our model can now study by looking at them, and then we can have like multiple of cells so that it can train really well. And then later on, we feed our data to the model, and then we can have now our unknown cell type could be annotated with the proper cell type. So now that we have some kind of idea, about what I do in a daily basis, and I would like to walk you through how I designed my experiment and want to share with you my key result. And first, I need to dissect the mouse cortex and extract cells from it. And then I will grow them on a dish with their favorite food. And we call this system a cell culture system, and which is a very powerful tool because we can study the effect of specific drug, or we could even model a disease on a dish. And then I grow them normally for 6 days, and at day 6, I treat one dish with activity blocker. So now you have to imagine we have two groups. So one group we call a control group without any treatment, and they grow just normally until the experiment. And we have the other group with an activity blocker. And why do I block activity? Because we know already that activity is really, really important for the neuron to mature and develop properly. And then at day 14, I perform first electrophysiology, and I notice something, then I do single-cell RNA sequencing. And with this experiment, I want to answer these two main questions. So which cell type is affected by activity blocker? And second, which mRNAs are changed in that particular cell type which are changed? So this is now, again, the typical part. So now we also, again, have the UMAP, and we see that, again, the individual point represents a cell, and they are grouped together based on their mRNA expression. And when they have a similar expression, they are grouped together and they have a same color. And here you see that 13 different color. Are you finished counting? So we will have 13 different color, meaning that 13 different cell type identified from our mouse cortical cell culture system. And like I said before, I'm interested in inhibitory neuron, the conductor cells. So here you see the 3 different type of inhibitory neuron, and here one. So we could identify 4 different inhibitory neurons in our system. But before I zoom in and look at their mRNA, I want to show you the image, how they look like, because it's more fun to watch. And now on the left, again, they are in the control conditions, so they grow normally for 14 days. And on the right, they are with activity blocker at day 6 from the day 14. And you see this little blob here, these are the cell body, and they have this fiber-like structure. These are the processes they are making to be connected to one another. And now if you were to compare this to image, you see clear differences. The first is that these cell bodies are so much smaller. And second is that they are making really, really less connection to one another. So with this result, we can claim that the blocking the activity is interfering the maturation and the development of inhibitory neurons. And now we want to zoom in again and know, but which inhibitory neuron specifically is affected the most? To answer this, we have to zoom in and look at the mRNA again. And here is again the UMAP, and we could, like I said before, we could identify 4 different inhibitory neurons. And this is when I use cell type, so different cell type as my observation, but I could have condition as my observation. Because we have 2 different conditions, so these are exactly same plot, so they are plotted based on the mRNA's expression, and individual point represents a cell. But this is when I use the cell type as observation and here as condition. So since we have 2 conditions, we have 2 different colors. And blue dot means here is the control condition, and the pink represents with activity blocker. And the way we look at this is actually quite simple. So we look first the purple, it should be purple, but the purple group, and we compare how this, inside of this purple group, how this blue and pink dot, how they're close or how far away they are. And you can see that in VIP group, that VIP sounds a little bit strange, but in the VIP group, the blue dot and the pink dot, they're kind of like intermixed quite well. And this is also true for the SST group, the orange group here, and then also the other group. But when you look at the pro-albumin group, the green group, you clearly see that the blue dot and the pink dot, they're quite separate, and they are not mixed very well. But what does this mean? This means that blocking the activity affects severely on this specific inhibitory neuronal type, which is para-albumin neuron. And now we can look at the mRNA that are actually changed with this, within this cell. And so here again, the blue dot is it's less expressed because of the activity blocker, and then here is more with the activity. But this is not so important to understand. The important message here is that we could identify really hundreds of mRNA that are changed in these pro-albumin neurons, and these mRNA are important for synapse formation and neuronal maturation. So with this result together, we could say the activity is really important for the para-albumin neuron to mature and grow properly. And we could even identify what are the mRNA that are necessary for this neuron to mature and develop properly. And our result is really, really exciting because para-albumin neurons, they're associated with many kinds of brain disorder. It is linked to Alzheimer's, epilepsy, schizophrenia, and autism spectrum disorder. But you might still wonder, so what? Why does this matter? Seulkee, I thought you were curing Alzheimer's, but you're not. But to really find the treatment plan, we have to understand the brain at a fundamental level. Which cell type are involved? What mRNAs are they expressing? And how do they change in different contexts? And by answering these questions, we lay the foundation to understand brain in health and disease, which would lead to the meaningful progress in the field of neuroscience. And just like brain needs diversity, we have also people in our group with diverse expertise. And I would like to thank you everyone in our group, and I would like to also thank you for your kind attention.
Thank you, Seulkee, and please keep on applauding. Thank you, Melissa, for your wonderful talk. Thank you so much. I would like to open the discussion session, and I'm quite sure that you have some questions in your head, but at the moment I have the microphone, so I want to start with the first question. The fact that blew my mind was the sheer amount of differences, 3,000 cell types. Do you think that number will hold during the next decades, or do you think that maybe in 10 years we meet again when we talk about 30,000 or 300,000 cell types? I don't know.
Well, Seulkee and I were actually just talking about this exact issue earlier when we were sitting outside. And the question is really, as the resolution gets better and better, and you can see more and more genes, it's quite possible that we will see more and more differentiation of these cells and these cell types. But the real question is, at what point is it really meaningful? So it could be that at some point the differences become so small that they're not really relevant functionally. But it's going to take some time for us to be able to catch up with how we can tell the functional effects of these cells, how they are behaving in the network, and how that relates to their transcriptional profile.
And also the process of taking a cell out of the tissue, putting it in a culture dish is a quite brutal method. Doesn't it change the cell?
Yes, it does. And you can now imagine like cells in your brain, and they will probably be very different. But we are doing this also to reduce the amount of the animal research that we have to do if we have to use our brain to start with. And just imagine, because we are growing them on a dish, we can treat them with different drug, and we could model things very easy way. If you were to treat them with drug, and in a live mouse, it's like a little bit difficult, and it has to go to the brain. So I think even though cell culture has its drawback because it's a very, very different system, but still it holds a lot of potential because we can answer really diverse questions with this system.
And then, Seulkee, you said something that made me wonder, is that with this wonderful metaphor with a cookbook, and that the mRNA is the recipe that the cell uses at a given time. So I wonder if what you look at when you see all these diverse expression patterns and the mRNA patterns, isn't it the one cell having the breakfast and the other one having the lunch, and maybe you just catch them on the wrong moment. Maybe an hour later the profile would be different. Would that be possible?
I think there are the key mRNAs that don't really change. So there will be like the housekeeping mRNAs, and there will be like this, like they're vegetarian, but they're trying, different food, they're trying the meat for the first time, but still it will be okay because we have a mathematical and statistical method to reduce this, what we call as a noise.
Are there first questions from the audience? Dare to raise your arms, otherwise I will keep on talking. Okay.
Thank you very much, Melissa and Seulkee, for the great presentation. I have a question to Seulkee. You mentioned when you explained your lab process that you feed the brain with its favorite food. So what's the brain's favorite food, and can I use it to grow my own brain?
Oh, that's actually a really good question. So, I mean, they like what we actually like as, you know, like as a whole organism, let's say. They like sugar, so they need sugar, and they also need, you know, they cannot have some kind of like germs or, you know, so they need to also take some antibiotics. So there are like mixture of different things that are in this food. They also have ions and yeah. But I guess like mainly sugar.
Wouldn't that cell culture medium be a good beverage after sports? I mean, would it give you all you need?
Yeah. Okay. I will try that in for my postdoc position.
I wonder what your boss would look like, because that stuff is quite expensive when you come into the lab and you see Seulkee. Okay, more questions from the audience. Yes, please.
When do the cell types start to differentiate? Is a mouse born with this 3,000 or maybe more types, or when did it start?
That's also an excellent question. A lot of the cell types are defined in the mouse before they're born. So we're using cultures from neonatal mice, so very, very newborn. And the general cell type, at least, seems to already be determined. It's interesting because there's quite a lot of work that's been done, not by us, but by other labs, where they think that it could be that the excitatory neurons, and we're talking about the cortex, this is one specific area of the brain, that those cells, the excitatory neurons there, may be more defined by the time that they migrate. So they start as progenitors, they migrate up to where they're supposed to be, and they're sort of defined by the time the mouse is born. And that the interneurons that we're looking at, they have a general phenotype or general identity, but as they get into their area where they're going to do their work in the network, their subtype becomes more refined. So with activity, which is actually also what we are seeing in the culture, this seems to be preserved, and what I'm referring to actually happens in the animal. So with humans, I'm not actually sure exactly. I don't. Maybe we don't really know, honestly, in the human brain. At one point, the cell types are fully defined, but we do also work with another experimental model, where we use actually human cells, and these are cells that they're cell lines that are generated originally from fibroblasts, from skin cells, from humans, and you can reprogram them into stem cells, pluripotent stem cells that can become really any type of cell. And then we drive them to become neurons by having them express certain factors. And so we have some idea of how, at least in a dish, we can watch neurons mature, human neurons mature, and make synapses and study their properties. But it's a very, very slow process, and they're never reaching the complexity of cell identity that you have in like an actual human brain. But excellent question.
More questions? What I wondered, you showed this slide, Seulkee, at the end, where you said that so many diseases may be affected by cell identity. Would it be oversimplified to say this cell type is responsible for disease A and this cell type is responsible for disease type B? Can you say that?
I think, you know, like brain is really, really, really complex organ, and they, so like for, let's say, in case of epilepsy, it could be the excitatory neurons are just overexciting. it could be also that inhibitory neurons are not functioning properly. They're not inhibiting enough the excitation. So what we see and what we study is just some aspect of this disease, but we cannot study the whole, like as disease as like whole system, you know. So I think it's difficult to say this one cell is like, you know, like affected in the whole disease, but it's just some aspect of disease is associated with this particular cell type. And this is actually really exciting news because this neuron is really, really special. And they're not just conductor, they're like real conductors. So they're like really inhibiting really at a very fast pace, and they have also very much energy demand because they have to, you know, have this fast activity. And yeah, I think since we know not just like the normal this paraalbumin neuron, the mRNA, but we also know like a little bit less developed and you know something is wrong with them. We also know these mRNA, so we can have this. Manipulate these mRNAs to study like something more into it. That's why the basic science is so important in this regard.
Thank you. If you want to have ask questions, and I just keep on talking, you can maybe just shout in between. I have a question. Yeah, there's a question.
Thank you for the talk. Why are we looking at mRNA instead of the final product protein?
Yeah, so this is actually a really good question. And the reason is actually really simple. Because we don't have the technology yet to really get the protein at the single cell level. But mRNA are just like really a lot of them exist, and we can amplify them with using this technique. We also know the PCR, you know, with the vaccine and the COVID. So the reason that we cannot use the protein is because of that. But we can like use different technique maybe to identify cell type when they are in the tissue level. But with really in the culture system, I think we're not there yet to use protein as the identifier of the cell type.
And you also said this at the very end, where you showed us that the activity level more affects the inhibitory neurons and not so much the excitatory neurons. That made me wonder, because in a brain, everything is interconnected, right? The excitatory neurons excite the inhibitory, inhibiting the excitatory. So can you there actually make that distinguishment? Isn't it just all reduced then? And I don't know, can you say the one is more affected than the other in an in vivo system? I mean, in a living organism?
Yeah, I think that it probably depends on the state of plasticity that the cell has and when this activity is stopped. And in our experiments, we do see effects in the excitatory neurons as well. And but in the in vivo situation, it may just be that by the time the animal is born and activity is really introduced into the network by synaptogenesis and stuff, that the cells are sort of more hardwired in the excitatory, in these at least these principal output neurons, than it is in the interneurons that still have a bit of plasticity and in their cell type, and their refinement can still be happening based on where they're needed and how their physiology needs to suit their function.
Mm. And then also one more question. Bitte? War hier eine Frage? Ah ja, bitte. Okay, dann.
Yeah, thank you very much. So you are doing the test with mice, and I was wondering how and to what degree you can apply your results to humans, especially when it comes to human diseases and human medicine. Thank you.
Again, it's a really great question. I mean, the kinds of experiments that we're doing just wouldn't really, to this extent, be possible with human tissue. Not yet. As I said, we are trying to use these induced human neurons, and eventually we may, with that, be able to get to a similar stage as what we see here. But I mean, in general, a lot, if not most, I mean, really most, of the preclinical work is still done with animal models. And as it turns out, we really can learn quite a lot from those models to then be able to look back at the human system and apply some of these things. I think that it's, again, very, very interesting that, you know, we see this cell type that seems to really carry a bit more of the burden, as we could say, of this treatment, this parvalbumin neuron, which is also seems to be associated with all of these diseases. So if we can then say, all right, well, which kinds of genes do we see that are changed here? We can now, using especially AI and the kinds of bigger data analyses, look into the human sequencing studies. There have been post-mortem human sequencing, single-cell sequencing studies that have been done. And we can say, all right, well, are the genes that we see now associated with some of these diseases that have been shown? You know, we can do these genome-wide studies, analyses, and look. And then we really might be able to apply directly some of these results to what we see in human disease. So I think it is applicable, but we also, always have to take things with a grain of salt, because there are differences between human and rodent neurons. We're learning this more and more now. Like, this is really an active area of research, but it's a good starting place because it is the only starting place.
We spoke about the role of activity in development of the brain cells. Is it really on a cellular level, or can you also transfer this to the growing of a child? I mean, how much activity, physical activity, does my boy need to mature? Is it the wrong comparison?
Yeah. So anyway, when we talk about activity today, it's what's happening in the brain. So I mean, like, if you move around more, yeah, probably it will help. But it's just like if you drink coffee, one cup of coffee every day, it'll make you healthier. So it's something more like this. I think the reason the brain needs activity, it's really in the level in the brain, but how this activity is generated and like this, I think it's a little bit different question.
One more question. Wonderful. There in the back.
Thank you for the talk. It was really interesting. I don't really know how to put this, but it's a little bit off topic maybe. I'm interested in AI, of course, because everyone has to use it now. What is your thoughts on things like ChatGPT or whatever models you are using on large language models? Are you afraid? And maybe as a second question, what's the first thing that you prompted, that you put into it?
Great questions. Well, I mean, this is my best friend. ChatGPT is like my best friend, my best colleagues, and it helps me a lot to code and to decode and so on. And I think, still like the researchers like us, like scientists, we will not at extinction, hopefully not. But if we don't know how to use it, how to use AI to ask questions questions that require a lot of data, that we call like big data, I think we cannot really do and understand these big data by just looking at them, because we don't have the capacity. I think the more the researchers or scientists like us use it, I think the greater the science it will get, I guess. And I mean, the model that I'm using is not a large language model, but I will not, let's not go details of it. But I think there was this saying, like, the ChatGPT will not take your job away, but the person who can use ChatGPT will do. So I think this is a good message to, I think you should now or go home and log into ChatGPT and start ask questions.
What a nice fire comment for Berlin Brain Session. Okay, stay for a minute. I just want to say thank you that you all have been here. Please, another warm applause for the wonderful talk of Melissa and Seulkee. And keep on putting your hands together for Felix on the camera and Jürgen for the technique. Thanks also for our sponsors, the Einstein Center for Neuroscience, and Linda and Claudia. Thanks for being here. You can stay a bit longer and discuss with Melissa and Seulkee, but we just have to close the official part. So thank you all for being here. Come again tomorrow and the other days for the microscopy days here in the Zeiss Großplanetarium. And when you go out, you pass by the little Schränkchen. Please take home one of these. There are more cool, one wouldn't believe it, but more cool events in Berlin, not only Berlin Brains. The new thing that we have is the excellent pub quiz. The 7 clusters of excellence put together all the energy and invite you once a month in the Fahimi bar in Neukölln. And now you learn so much about neuroscience. Go there and show off a bit and test your knowledge in this excellence pub quiz. Take one of these flyers back home. Thank you all for being here. Bye. See you next time.